In this module, we will be discussing the electrophoresis of nucleic acid. These are the techniques which are used to separate or to identify the different nucleic acid present. Remember that previously we have been discussing the electrophoresis of the proteins or the nucleotide. Now, electrophoresis for DNA is actually carried out by the agarose gel, or on the agarose gel, the separation of majority of DNA molecules is carried out. Now, DNA molecules and their fragments, they are larger than the protein molecule. And most DNA fragments that would be unable to enter a polyacrylamide gel. The larger the pore size of an agarose gel is therefore required because DNA molecules are larger molecules. Now, nucleotides are larger molecule, and when talking about single-stranded DNA, it is common to refer to size in terms of nucleotides, which is abbreviated by NT. And the separation in agarose gel is achieved because of resistance to their movement caused by the gel matrix. You see, the DNA molecule is there, and there is a gel matrix which is there, and then the separation is due to the resistance of that matrix and the DNA molecule. They are separated. The largest molecule will have the most difficulty in passing through the gel pore. It is just like the sieve, as where the larger particle or the larger molecules are retained and other smaller molecules or the smaller size DNA is, as a matter of fact, is allowed to pass. The smallest molecule, therefore, they will be relatively unhindered. They will pass through easily through the gel. The mobility of DNA molecules during gel electrophoresis, that will depend on size, as already pointed out previously, and the smallest molecule, actually, it moves faster. The molecule, if it is small, it will move faster in that gel and it will go above. The gel concentration, in this case, that must be chosen to suit the size range of the molecules to be separated. Just like this, that if we have to separate different molecules or different particles, we select different sieves of different particle or different size. Similarly here, the gel concentration has to be chosen so that they are actually appropriate or they are suitable to the size of the molecule with which we are working. Now, DNA gels, they are invariably run as horizontal, submarine, and submerged gel. So they are horizontal, submarine, and submerged gel. That is, they are dipped in the buffer and in the horizontal shape. Now, just as the photograph, this shows 4 tracks of the gel, and in this case, it is 0.8% agarose submarine gel, where the DNA molecules, they are being separated, and they are being separated according to the size of their particle. DNA sequencing gels, they are, as a matter of fact, agarose gel electrophoresis of DNA. And that is the DNA electrophoresis gel. They are used commonly, frequently, and serve as the workhorse technique for a molecular biologist, because the molecular biologist has to separate the DNA molecule for frequently. A different form of electrophoresis has to be used when DNA sequences are to be determined. That we have to determine the DNA sequence, that where are the nucleotides and how they are organized in a molecule. The final analysis, it usually involves preparing single-stranded DNA. First you prepare single-stranded DNA molecule, and that should be shorter than 1000 nucleotide units and different in size by only one nucleotide. So you see how precise it is and how to small unit we can go. Electrophoresis in agarose, that can be used as a preparative method for DNA analysis. That is, for DNA analysis, we have to use the electrophoresis method. Now, there are other methods like pulse field gel electrophoresis. Now, in this case, the method basically involves electrophoresis in agarose, where two electric fields, they are applied alternatively. Two electric fields, they are applied alternatively at different angles for defined time period, such as 60 seconds. So those electric field, it is there and it moves or it changes for 60 seconds from one angle to another angle. Now, pulse field gel electrophoresis is used for identifying the course of outbreaks of bacterial foodborne illnesses, such as the illnesses which are caused by Salmonella typhi, that is the typhoid and other diseases. Example is the DNA from Salmonella species, when digested with the restriction enzyme X-ball, gives around 1.5 fragments. They are ranging from 25 kilobases to 680 kilobases. So that is the difference between them. Just as we have shown here that this is the pulse field gel electrophoresis of a digestion pattern of the DNA. Now, electrophoresis of RNA, like DNA, electrophoresis of RNA is usually carried out in agarose gel, and the principle of the separation is based on the size, and it is exactly the same as for DNA, as shown in this slide, where the separation of yeast RNA is now 1.5% gel agarose. Now, that is the rapid method of checking the integrity of RNA is by electrophoresis on a 2% agarose gel in about 1 hour. So you see now we are increasing the concentration of agarose gel. Now, when we increase the concentration of agarose gel, what happens? We are decreasing the pore size of that, because the concentrated the agarose gel is, the smaller becomes the pore. And the ribosomal RNAs, that is 18S and 28S, which are commonly taken as the identification for the microorganism, they are clearly resolved, and any degradation seen as a smear or DNA contamination, that can be seen easily. So it means that with these methods, they give the microbiologist a handle to differentiate, to separate, and to study the molecules of DNA and molecules of RNA. And they are extremely useful techniques in molecular biology.